Hello everyone. Today we are going to start a new lecture series where we'll be learning some important aspects related to the control of rice weevil or the Pseudophilus oryzae, which is a stored grain pest. So I would like to start learning about that. So we'll be reading different research papers related to this topic. So I'm using this platform as a you know mm, record my study progress. So that's the purpose of this series. So tropical medicinal plant extracts against rice weevil Pseudophilus oryzae. It is a study paper. So we will be learning about that. So rice weevil or Pseudophilus oryzae is one of the major pests of stored commodities. The adult weevils feed on rice and the females lay eggs inside rice kernels. In the study, chemical composition of extracts from black pepper, Pippa nigrum L and Physic nut, Jatropha caucasus were tested against this pest. So black pepper was used. Black pepper. Okay. Along with that, Jatropha curcus were tested against this pest under laboratory conditions. The chemical composition of the extracts were identified by gas chromatography and gas chromatography Y mass spectroscopy GSMS. The major extracted components of P. nigrum were piperin and caryophyllene, and for the other one, that is Jatropha, were oleic acid, linoleic acid, and palmitic acid. The mortality of adults increased with increasing concentration of extracts from 2 to 10 ml per gram. They used about 2 to 10 ml per gram of rice. Okay. And exposure time from 24 hours to 72 hours. The petroleum ether LC50 1.61 ml per gram and chloroform extracts LC50 1.70 ml per gram of Pepper showed higher mortality rate compared to petroleum ether extracts. So here we will be performing petroleum ether extracts of all these plant materials. And this petroleum ether extract showed 99.56 to 93.56 and 66% mortality respectively. The P. nigrum extracts were significantly repelled. So that is a discovery compared to Chickercas extracts. So petroleum ether and chloroform extracts. So they performed petroleum and chloroform extracts. And I think the chloroform extracts definitely kill the, I don't know, but still. Petroleum ether extracts of these extract also showed strong anti-fedent and opposed to progeny production. So furthermore, F1 adults were suppressed at the lowest concentration of 2 ml per gram and no F1 was produced in all treatments. The results of this study show that pepper nigrum and J. curcas extracts were able to protect stored grains. Okay. So stored agricultural and animal origin products are attacked by more than 600 species of beetle pests, 70 species of moths and adults, and about 355 species of mites, which cause quantitative and qualitative losses. So the damage may lose about 5 to 10% in temperature zone and 20 to 30% in tropical zone. So one of the major pests of stored commodities in tropics is rice weevil, Pseudophilus oryzae, which is involved in the Coleoptera curculionidae. Adult weevils feed on rice and lay their eggs inside rice kernels where the larva can develop to adult stage. So to control of pests of storage, methyl bromide and phosphine are used. That may several problems on stored products. Okay, that create some problems. So therefore, alternative pest control method need to be developed. For example, a lot of natural pesticides can be diverted from higher plants. These products do not leave harmful residue. Okay, to the environment and have medicinal properties for human with lower toxicity to mammals. So black pepper and physic nut are examples of such plants which can possess medicinal, insecticidal, repellent or anti-ficant property. So P. nigrum is a perennial vine found in tropical regions and native to the Indo-Malaysian region, whereas J. curcus is a perennial tree found in tropical regions such as India. J. curcus extracts were reported to be a toxic toward leaf-cutting larvae. Oh. So in this study, we evaluated the insecticidal effects of those medicinal plant extracts to control rice weevil. Materials and methods, insect rearing. Before the conduction of this experiment, we need to have a, Cetaphil is a rice seed group with which we can conduct this experiment. So colonies of rice weevil were obtained from an entomology laboratory stock culture of University Putra, Malaysia, UPM, and reared on whole grain rice grains, initially 13 to 14% moisture content in plastic container under the laboratory conditions at 28 plus or minus 2 degrees Celsius, 75 plus or minus 5% RH, and L12 and D12. The subcultures and the tests were carried out under the same conditions. 7 to 14 days old adults of Cetaphilus oryzae were used to do the experiment. So, extraction of plant extracts. Plant extracts were prepared by the percolation method described by Sarkar et al. So, that percolation method by Sarkar et al. Okay. With some modifications, dry seeds. Oh, here seeds are used. Okay, dry seeds of this species and J. curcus were grounded to powder using a grinder prior to oil extraction. The powder were soaked with 95% of methanol for 48 hours. So powder soaked with 95% of methanol for 48 hours, filtered, and the residues were extracted afterwards. All filtrates were combined and concentrated by rotary evaporator. An equal volume of water added to the crude extract, and extraction were done by petroleum ether, and they used petroleum ether for extraction. Okay, so the residues were followed by chloroform and were collected as chloroform fraction and residues, aqueous extract, and finally were concentrated by rotary evaporator, 40 degrees Celsius, and put at 4 degrees Celsius. So gas chromatographic analysis was performed, extractions to determine the fatty acids using a 30 m into 0.25 mm ID. One microliter was injected by an autosampler into a chromatograph equipped with a split injector and a flame ionization detector. The split ratio was 1:20 after injection of 1 μl of the fatty acid methyl esters. The injector temperature was programmed at 250 degrees Celsius and the detector temperature was programmed at 300 degrees Celsius. The column temperature program initiated runs at 100 degrees Celsius for 2 minutes, warmed up to 170 degrees Celsius at 10 degrees Celsius per minute, hold for 2 minutes, warmed up to 200 degrees Celsius at 7.5 degrees Celsius per minute, and then hold for 20 minutes to facilitate optimal separation. The GC mass analysis was carried out to determine the major components of the extraction on the Shimadzu GCMS. plus equipped with a BPX-5 column. Helium as carrier gas in electronic impact mode and at a flow rate of 1 split injection ratio. The injector and the GCMS interface were kept at 320 degrees Celsius. The column temperature program was as follows: 50 degrees Celsius, heating at 10 degrees Celsius per minute until 320 degrees Celsius and remaining at this temperature for 15 minutes. The components of the oils were identified by comparison of the mass spectra with the NIST NOT8 library information. So then next one is toxicity tests. So we can also add a portion where we will be showing toxicity tests. So toxicity effects of plant extracts on Cytophilus raceae were carried out in laboratory according to the methods of I'm not sure about the pronunciation, Talukdar and House in 1994. Toxicity test. Okay. With some modifications, 5-gram rice were soaked in. So how are they done? Just listen. 5-gram rice. 5-gram rice were soaked in a 1 ml of 1 ml of 1, 2, 3, 4 and 5% extractions. 3 fractions and another one. Subsequently, then the control rice was treated with solvent only. Okay, control with solvent. So whatever you are using, use as a control. So control with solvent only. Okay, and air dried. Then we will be air drying for 30 minutes to evaporate the solvent. Oh, that's how they conducted this. Evaporate the solvent. Okay, then each sample were placed in a vial and 10 adults of Cytophilus raceae were released on each of them. 10 adults on each vial. Okay, and the experiment were done with 5 replicates. So 5 times repeated. 5 times they repeated, and this is going to be really important. 5 times repeated. Okay. So then the adult mortality was recorded after 24, 48 and 72 hours. The LC50 and LC95 values of 6 extracts were calculated by Probit analysis using Polo Plus software. We will be using a software. Probit analysis by 1971 using Polo Plus software. The original data were corrected by Abbott's formula. Abbott's 1925 formula transformed into percentage and square root values, and then variance analysis ANOVA were done by SAS program. In the end, mean values were adjusted by . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . multi